Okay, hello again. I've got a little review here for the reproductive system portion of Unit 5. I've got some example questions and we'll go back over some of the diagrams. I'm not going to hit every topic from the reproductive system in this review. Instead I'm going to try to home in more on some of the more difficult concepts, and for most students that revolves a lot around the ovarian cycle and the uterine cycle and the hormones and what happens if a pregnancy does occur, what happens if a pregnancy does not occur, and how all that works. So we'll spend a fair amount of time there. Of course, we'll also go through sperm cell production in males as well, even though the story is not quite as complicated on the guy side of things. All right. So first of all, let's hang on just a second. Okay, never mind. Thought I was going to have an interruption. First of all, let's talk about meiosis and review a little bit about meiosis. Remember, meiosis is the special process that's used during oocyte and sperm production to help. You've got to reduce the number of chromosomes in the nuclei of those cells from 46 to 23, so that when sperm meets egg, 23 chromosomes from the male, 23 from the female get together, and now you're back to 46 for that first cell that occurs in the new individual, in the zygote that forms. So remember, when you're looking at these diagrams, 2n for humans is 46, and that means you've got two copies of each chromosome. And n refers to 23. That's another term for that is haploid. For this, another term is diploid. All right, and so if we review over on the oogenesis side of things, get on my diagram there. Now the story gets a little more complex than this, but in the ovaries you have a cell type. These are big cells, big individual cells called primary oocytes, and they're 2n. They've got 46 of the mama's chromosomes in them. And then at a point just before ovulation takes place, the primary oocyte divides and becomes a large secondary oocyte. It undergoes meiosis 1. If you go back and review meiosis 1 and 2. And the other cell, this cell divides into two. The other one is a little small cell, which is really just a nucleus with a little cytoplasm around it, and that's called a polar body. So if you guys remember, this is a haploid cell, the secondary oocyte. It has 23 chromosomes, but they're duplicated. So each of those 23 is actually a duplicated chromosome. That's when they're drawn like an X. And when meiosis 2 occurs, those duplicated chromosomes separate into individual cells. And so that's when you're really down to having just 23 chromosomes in the final cells there that form. All right, over on the sperm cell side, you have primary spermatocytes that have 46 chromosomes in them. They're diploid. And each of those undergoes meiosis 1 and becomes two secondary spermatocytes. Each of those undergoes meiosis 2 and becomes what's called a spermatid. So those are still round-shaped cells, but now they just have a single copy of each of the 23 chromosomes from dad. Each of those then undergoes, there's no more cell division, no more meiosis taking place, but each of these undergoes a big-time change process and takes on the characteristics that we recognize of sperm cells, with the head and the midpiece and the tail, so that they can swim around and try to find an egg to fertilize. When the sperm meets the secondary oocyte, remember when a female ovulates, it's a secondary oocyte that actually gets released out of the ovary, starts traveling down a fallopian tube. Sperm meets the secondary oocyte, that's fertilization. And it's actually only when the sperm cell enters the secondary oocyte, then that nucleus in there, that secondary oocyte, actually completes meiosis 2, which is kind of strange to think about, and that leads to another polar body. And now the DNA from the sperm, the chromosomes, the DNA, the chromosomes from the mature ovum here get together and now we're back to 2N. We have 46 chromosomes, 23 from mom and 23 from dad, and that forms the zygote. That is the first cell of the new human individual that has been formed. All right, so here's an example question. This is one of those select all that apply. Remember, with those, kind of just go down the list of answers and treat each one like a true false. Select each of the following cells that is diploid. All right, spermatozoa. Spermatozoa is another term for a mature sperm cell. So is that going to be diploid? Hopefully not. If it is, and it winds up fertilizing an egg, that zygote that forms from that will have way too many chromosomes, and it's not going to be viable. It's going to die. How about the oogonia? Yeah, those are diploid. If you go back and look, those are primitive cells that occur before the primary oocyte, so they mature into primary oocytes. And since primary oocytes are diploid, those oogonia are going to be diploid as well. How about a secondary spermatocyte? No, we just talked about how those are actually haploid. Cardiac muscle fiber. Yes, that's just a somatic cell of the body, one of our body's cells, and those are going to be diploid cells. They're going to have 46 chromosomes. How about a zygote? Yeah, we just saw that's a single diploid cell. And how about a sustentacular cell? That's one of those cells in the wall of the seminiferous tubule in the or a cell in a wall of a seminiferous tubule in a testis. And yeah, that's one of the body's cells, and so that's going to be a diploid cell as well. Your only haploid cells are these mature sperm, or these cells that are involved in sperm formation and egg formation. Otherwise, the cells in your body are going to be diploid. Now, there are some exceptions, like your red blood cells. Mature red blood cells don't have a nucleus, so they don't have any chromosomes inside them. All right, a little bit of a closer look at spermatogenesis, and this picture should look familiar to you. This is actually some stained tissues from the testis of a male, and what you are seeing here, right in here in cross section, that is a seminiferous tubule that has been cut open, and so you're kind of looking in on the end of it. So right there, that's the lumen of the seminiferous, of a single seminiferous tubule, and all in through here, this is where you have spermatogenesis taking place, the formation, all these meiosis stages that lead to sperm cell formation. And then over here, you would have another, the seminiferous tubules are all wadded up, kind of like wadded up pieces of spaghetti. And so there's another one portion of it over here. Now, in between these seminiferous tubules, you have these interstitial spaces, and in there you have interstitial cells. And the thing you need to remember about those guys is they are the cells that make testosterone in the male. Those are your testosterone-producing cells. Okay, and then more of a cartoon-type figure, what's going on. So we're looking at an area like this in the wall of a seminiferous tubule. Out toward the edge, you have these spermatogonia. Spermatogonium is singular. That's one of those. And those are diploid cells. Those are cells that are, as they divide and mature, they're going to become the cells that undergo spermatogenesis. So a spermatogonium divides into a type A daughter cell and a type B daughter cell. The type A's continue to hang out in the near the edge of the seminiferous tubule, and they just continue to offer a supply of cells that can divide and go through this sperm formation process. The type B daughter cells, now that cell division, by the way, spermatogonium going to a type A and a type B, that's mitosis. So you're going from a diploid cell to two diploid cells. The type B daughter cell undergoes some changes and becomes a primary spermatocyte. So remember, that's a 2N diploid cell. One of those divides into two secondary spermatocytes. And they're considered haploid, but keep in mind you've got 23 duplicated chromosomes in there, so they're attached to each other like that. And then each of those divides into early spermatids. That's your meiosis II process. So those duplicated chromosomes separate each individual into a separate early spermatid. And then those early, meiosis is now over. The rest of your sperm formation involves those early spermatids maturing into the cells that we see down here that look more like what we're used to seeing as diagrams of sperm cells. And these are going to be in here. They're in the lumen of the seminiferous tubules, and then they're able to start moving from there out of the testis and into the epididymis as they start their journey toward the outside of the body. All right, the hormonal control, just to review this in the male. So the hypothalamus makes a hormone, gonadotropin-releasing hormone, GnRH, and that travels the short distance to the anterior pituitary, and it triggers the anterior pituitary to make there are cells in there that make FSH, follicle-stimulating hormone, and LH. LH acts on those interstitial cells in the testes, and it triggers them to produce testosterone. And of course, testosterone has many effects. That's the hormone that's primarily responsible for the development of the secondary sex characteristics in males. So all the male type features of the body are triggered by testosterone. All right, FSH, this part of the story is a little bit more difficult to grasp. FSH actually works on the sustanacular cells. Those are the target, and it stimulates those cells to make a protein called ABP, androgen binding protein. Testosterone is a type of androgen. Androgen refers to male sex hormone. And so the ABP is kind of like a magnet for some of this testosterone, and so it keeps the testosterone concentrated around this area where all this sperm cell formation is occurring. And by doing that, it's stimulating spermatogenesis. So you got to have the testosterone being made to stimulate spermatogenesis, and you got to have the FSH telling those sustanacular cells to secrete androgen binding protein, because that's functions kind of like a magnet for testosterone and keeps some of that testosterone concentrated around the walls of the seminiferous tubules, so that spermatogenesis will wind up getting stimulated. Okay, then our negative feedback. Notice testosterone has negative feedback up here on the anterior pituitary and will lower the amounts of FSH and LH. And inhibin is produced by the sustanacular cells and plays a similar role. So you've got negative feedback that takes place to control the levels of testosterone inside the body, and also to help regulate the amount of sperm being produced in the male. All right, here's one of those annoying multiple answer. I have a lot of multiple answer questions in this review. I don't want you to think that the test is going to be overloaded with multiple answer questions, but they're just really good to use for review. And like I said, I tried to put some more challenging type questions in here for the review to help us go through the material. All right, select each of the following that is true about spermatogenesis, sperm cell formation. All right, A, go through the list, treat it like a true false. FSH stimulates production of testosterone. No, LH tells those interstitial cells, sorry, I was blanking out there for a minute, to produce testosterone. All right, sustanacular cells are not required. That's false. So we just talked about how they make ABP, which helps keep the testosterone concentrated in the walls of the seminiferous tubules so that spermatogenesis can be stimulated. And they play other roles as well. Go back and check and look through the different functions of the sustanacular cells that we talked about. C, sustanacular cells secrete ABP, which binds testosterone. Yep. LH stimulates testosterone secretion. Yes. Inhibin secretion directly inhibits testosterone binding to ABP. See, that one might sound kind of reasonable when you're first reading through it. This is why you got to have to kind of know your stuff. And no, inhibin functions up at the anterior pituitary level and tells the anterior pituitary to quit making FSH, quit making as much FSH and LH. LH binds receptors on early spermatids and stimulates spermiogenesis. So do you guys remember what that term is? Because that sounds very similar to spermatogenesis. Spermatogenesis is describing this whole process of going from the spermatogonium to primary spermatocyte, secondary spermatocyte, early spermatids, etc. Spermiogenesis is after you have the early spermatids, so the meiosis steps are over, those round cells going through the dramatic changes so that they look like sperm cells, so that they have a head and a midpiece and a tail. And that's the spermiogenesis part. And no, that was not a function of LH that we discussed. LH stimulates the interstitial cells to make testosterone. GnRH is important for spermatogenesis. Yeah, that's made by the hypothalamus, but that is a releasing hormone that stimulates the anterior pituitary to secrete FSH and LH. Okay, so C, D, and G. Okay, poor Pablo, he has a destructive tumor of the anterior pituitary that has destroyed the cells that secrete gonadotropins. Do you guys remember what gonadotropin? That's another term that's referring to FSH and LH. Gonadotropins are going to be hormones that act on the gonads, so ovaries in females, testes in males. What is the likely outcome for poor Pablo? So this is not a multiple answer. He's not making LH and FSH, so will he have excessive production of testosterone? I don't think so, because he's not producing LH, and LH is required to stimulate testosterone production by the interstitial cells. Low testosterone production, low sperm cell production. Yeah, most likely, if he's not making FSH and LH, you got to have those two hormones to trigger that to take place. Acromegaly, blast from the first part of the semester. No, that is stimulated by overproduction of growth hormone after the growth plates have closed. High levels of testosterone but low sperm cell production. I don't think so. He's not going to have high levels of testosterone because he's not making the LH that stimulates that to take place. So our answer there was B. All right, true or false? Sperm cells are fully functional and motile once they enter the lumen of a seminiferous tubule. Hopefully you're thinking false. Go back and review the information that we discussed about when sperm cells become good swimmers. It's not anywhere near that early. They continue to become better swimmers as they move into the epididymis and then on forward from there. So go back and review that information in your notes as well. Okay, this is kind of an odd diagram from your textbook. It can take a minute to realize what kind of view you're getting here, but this is a posterior view of, now hopefully you recognize that that's the bladder, and of course there's a testis, and epididymis, vas deferens, reductus deferens, and then that's wrapping around the top of the bladder, and this wider area there is the ampulla of the vas deferens. Hopefully you recognize the little yellow gland over there. That's a seminal vesicle, which produces a large amount of the fluids that are present in semen. And then right in here, where those two come together on each side, that's where you have the ejaculatory duct. So sperm combines with the fluids from the seminal vesicles, and those two things are combined and released during ejaculation. And as ejaculation takes place, that fluid enters the urethra, the prostatic urethra, where it's passing through the prostate, and there are lots of little pores in the wall of the urethra there that allow fluids made by the prostate to also combine with the semen. So that's going to be taking place during ejaculation. And of course, the semen then travels down the spongy urethra in the penis and on outward to the outside of the body. All right, so are sperm cells stored inside the seminiferous tubules prior to ejaculation? No, the sperm cells that are going to be ejaculated are going to be up in that ejaculatory duct area over here. On each side, you have two ejaculatory ducts, and those are formed from the union of the ampulla, the widened part of the vas deferens, where that meets the seminal vesicle. There's a duct connected to the seminal vesicle, and those two things come together. So that's going to be false. All right, select each of the following that is true about semen content. Most of the volume comes from the bulbourethral glands. Hopefully you're thinking false on that. The bulbourethral glands secrete a fluid in advance of ejaculation, so it coats the inside lining of the urethra and makes it a more hospitable environment for the sperm cells as they travel through there during ejaculation, and also functions as a lubricant. All right, contains about 20% urine. I hope not. Urine is actually toxic to sperm cells, so you wouldn't want 20% urine in your semen. The volume is reduced by 50% following vasectomy. No, go back and review what happens during a vasectomy procedure. Vasectomy, the vas deferens on each side gets cut, but think about where most of the volume of the semen comes from. So a vasectomy is going to prevent sperm cells from moving from the testis upward and being combined in with the semen, but most of your semen volume comes from the seminal vesicles and the prostate glands. All right, does it contain fructose to supply sugars to the sperm? Yes, it does. If you go back and review, the fructose is a type of monosaccharide, and it provides energy to those sperm cells. Does semen have an acidic pH? I hope not. Sperm cells don't like an acidic pH. They can't swim well, and it's toxic for them. Does it contain prostaglandins? Prostaglandins are signaling molecules, kind of like hormones, but they tend to act in local areas to stimulate muscular contractions in the female. If you go back and look, that is one of the things that we talked about, semen containing these signaling molecules that actually stimulate the smooth muscle cells in the organs of the female to contract, and that helps propel the sperm cells forward after ejaculation. And G, contains enzymes that trigger coagulation after ejaculation. That's another thing. If you go back and look, semen after ejaculation coagulates or clumps together pretty quickly, and the idea there is it helps prevent it from leaking out of the vagina of the female, and it makes it increases the likelihood that fertilization can take place. So our answers there were D, F, and G. All right, moving over to the female side of things, the ovarian and the menstrual cycles. And first, let's take a look at what's going on in the ovaries. So these are all ovary events that we see over here. And let me turn off my pen because I think I'm just going to do this with the cursor if we can. All right, so one thing to keep in mind, some of the terminology associated with the oocytes and the follicles sounds the same, but so you need to pay close attention to whether a term is referring to the follicle, that's the multicellular structure that surrounds an oocyte and protects it, houses it, versus the oocyte itself, which is just a single cell. So over here on this left side, we're homing in on just the oocytes and what's going on during meiosis, and this more or less parallels what goes on during sperm cell formation in the male, but it happens with much, much, much less frequency. So in the ovaries, there are cells called oogonia. Oogonium is singular, And that's a stem cell, which means it's a source for additional cells, and it's unspecialized at that point. And that's a diploid cell. Those undergo mitosis and create primary oocytes. And these are the main functional cells in the ovaries. And so those hang out in the ovaries during infancy and childhood. And then each month from puberty to menopause, menopause is when the menstrual cycles cease, you will have a primary oocyte that undergoes meiosis 1 and divides into a secondary oocyte, and then the other cell is a polar body like we talked about before. And this is the cell, this happens right before ovulation occurs. Ovulation is when the secondary oocyte gets released from the ovary and enters a fallopian tube. And it remains in that state as it's traveling down the fallopian tube. If it gets lucky and a sperm cell penetrates it, then it completes meiosis 2 and becomes an ovum. Again, that's after the sperm cell has penetrated it. And then very quickly, the chromosomes from the ovum and the chromosomes from the head of the sperm cell are going to merge together and create a single-celled zygote. All right, so on this side, as we talked about, you only maintain these primary oocytes, secondary oocytes, and the ovum are very, very large cells. So when cell division takes place, these meiotic divisions occur. The other cell that picks up the other set of chromosomes is a polar body, which is a little wimpy cell that just goes on and degenerates or dies. All right, so over here on the follicle side, now the follicles again are providing the housing for these oocytes. And so don't confuse the terminology between the two. So in the ovaries you have primordial follicles where you just have a little single layer of cells surrounding the primary oocyte. So this guy right here is inside the primordial follicle right there, and it's got much smaller cells providing a covering, a single layered covering around it. All right, and then primordial follicles become primary follicles. Those cells of the follicle change shape and elongate. And then in response to primarily follicle stimulating hormone and also later estrogen, these primary follicles, some of them become secondary follicles, and hopefully just one per month develops into a mature follicle or a Graafian follicle, a vesicular follicle. So that's where it's very large, and inside there you still have a primary oocyte in there. So just because you're calling this a secondary follicle, that's still a primary oocyte. Secondary here is referring to the how mature the follicle is. So I know that can get kind of confusing. You just have to pay close attention to what's being discussed. And there is a fluid-filled space in there as well called the antrum. And here's ovulation. So again, that cell right here undergoes meiosis 1 and becomes a secondary oocyte just before ovulation. This is ovulation taking place. So this leaves the ovary and enters the fallopian tube, hopefully. All right, how about all these cells that were left over from this mature follicle up here, this vesicular follicle? They undergo changes and become a corpus luteum, which is a yellowish structure which plays a really important role in producing hormones that make sure that uterine lining stays nice and thick and producing nourishing fluids for an embryo if it winds up attaching. If no pregnancy occurs, this corpus luteum structure degrades and stops making the hormones that maintain the uterine lining, and then your uterine lining degrades and sloughs off, and that's when you have the menstrual period. All right, here's another way of looking at the ovarian cycle. A typical 28-day cycle can be divided up into the first phase. That's called the follicular phase. And again, that's where you have primary follicles becoming secondary, and hopefully just one secondary follicle matures all the way into a vesicular follicle and actually ovulates and releases the secondary oocyte inside into a fallopian tube. Hopefully just one per month. If you had two or more, that's where you could have um, multiple embryos developing. All right, after ovulation takes place, again, these leftover cells from the follicle, those are granulosa cells, they undergo changes and become this structure, corpus luteum, which this is still in the ovary. This is left over in the ovary. And this is producing estrogen and progesterone, which is very important for maintaining the inner lining of the uterus in a nice, thick, nourishing state, getting it ready for an embryo to attach. If no embryo attaches, though, it's going to degenerate, and it becomes it shrivels up and becomes this whitish structure, which is essentially like a little type of scar tissue. All right, and how about our hormone story over here? So this can get pretty complex in the female. So females also produce the GnRH, just like we saw with the males. That's coming from the hypothalamus. That stimulates the anterior pituitary to produce FSH and LH. FSH, follicle stimulating hormone, so its name makes more sense in females because it's actually stimulating follicles to develop and thicken, so convert from primary to secondary, or from secondary into a mature vesicular or Graafian follicle. And then LH, so remember LH in males is stimulating interstitial cells to make testosterone. In females, one of the things LH does is it's responsible for stimulating estrogen production. And it's kind of a little bit of a convoluted process. So you have some cells that are forming the outermost, they're connective tissue cells around the outermost part of the follicle called thecal cells, and they actually produce androgens, which are male sex hormones. And then your granulosa cells here take up those androgens and convert them to estrogens. So the main female sex hormone that's responsible for the secondary sex characteristics in female estrogens. And as these estrogens are made, and the reason it's estrogens, there's more than one type that are closely related, they act on the follicles themselves and trigger them to become even more mature. And again, hopefully you just have one becoming a completely mature follicle that's going to ovulate each month. Negative feedback, these estrogens have negative feedback up here on GnRH and FSH and LH. The granulosa cells make inhibin, like we saw also in the male, from the sustenacular cells. They also have negative feedback on those hormones being produced. And the idea there is that's going to hopefully prevent more follicles from maturing, and it's also hopefully going to help prevent more than one secondary oocyte from being ovulated out of the ovary each month. All right, however, there is a little complex part of the story here. As more and more and more estrogen gets made, eventually you reach a point where there's so much estrogen around that instead of negative feedback, it temporarily has a positive feedback effect up here on the hypothalamus and the anterior pituitary. And that causes a surge. There's a burst in production of LH right at the middle of the cycle, about day 14. And that is what triggers the mature follicle to rupture. Out comes the ovulated secondary oocyte. That's headed over into a fallopian tube, where it's going to travel down the fallopian tube and wait around and see whether it wins the jackpot and gets fertilized by sperm cells. And again, the leftover granulosa cells here from the mature follicle convert into this corpus luteum. And those cells in there make progesterone and estrogens. Those are important for maintaining the inner lining of the uterus. And they'll also produce inhibin. All of those things also have negative feedback up here at the hypothalamus and the anterior pituitary. And again, that is to keep things happening in a monthly cycle and to prevent, you don't want another oocyte maturing and being ovulated during a single cycle. So that negative feedback is important to ensure that things stay on the clock the way they're supposed to. This is a pretty good diagram which shows you a timeline for what's going on um, hormone-wise and in the ovaries and in the uterus and how all these things line up together. Again, you're looking at a timeline here from day 0 to 28 days, and menstruation is actually considered to begin at day 0, the first day, the very beginning of the next cycle. So that's menses or menstruation. That's where the lining that built up on the inside of the uterus the previous month dies, and the uterus contracts and sloughs that off. During that first 14 days, you have the growing follicles in the ovaries. At day 14, you get that burst of luteinizing hormone seen here in blue. These are hormone levels for FSH and LH. There's also a little burst of FSH there, but that triggers ovulation. There comes that secondary oocyte bursting out of the ovary. These leftover cells become the corpus luteum, and those cells are going to make estrogens here in yellow and progesterone here in purple during what's called the luteal phase of the cycle, that second half. That's referring to the corpus luteum being alive and functioning. If we follow our estrogen diagram, estradiol is the main type of estrogen, so that's what's being referred to there. But if you look at in the first half of the cycle, there's estrogen levels rise, and that's being produced by these cells in the follicles. The thecal cells and the granulosa cells in there are cooperating to make estrogen. And then at ovulation, eventually this triggers that burst of LH. Ovulation occurs. Estrogen levels actually drop off temporarily. But then as this corpus luteum over here forms, the cells in there start cranking out lots of progesterone, lots of estrogen again, and that's important for maintaining this lining, making sure the glands in here, the little white areas, are supposed to be the glands that will secrete the nourishing fluids to an embryo if it attaches. If no embryo attaches, the corpus luteum doesn't live very long. It just lives a few days and then it dies. Progesterone levels drop, estrogen levels drop. Progesterone suppresses uterine contractions. So if you don't have the progesterone around anymore, the uterus starts contracting and sloughs off the lining that was created. And then you're back over here at the beginning. You start a new round of the uterine cycle. So I know that's kind of a complex story, but just go back and review. What structures are making which hormones and when and why. Go back and, you should know what the purpose of a follicle is. What is the corpus luteum? Where is that coming from? Why does it need to make progesterone and estrogen? How long does it do it? What happens if there's a pregnancy? If there's a pregnancy, the early embryo is going to make something. Hopefully you're thinking there's a hormone that the early embryo makes. Hopefully you're thinking HCG for human chorionic gonadotropin. And HCG, that hormone being produced by the early embryo, acts on the corpus luteum and makes the corpus luteum stay alive. It doesn't die after just a few days. If there's a pregnancy, this stays alive, keeps making progesterone and estrogen, and that's important for the first several weeks of a pregnancy to make sure you maintain that uterine lining. If for some reason the corpus luteum dies even though you're pregnant, you're most likely going to have a miscarriage if you don't know that that's happening. Eventually, the placenta that develops makes progesterone and estrogen, and then you don't need that corpus luteum after the first several weeks of a pregnancy because the placenta is going to take over and make those hormones. All right, select each of the following that is true about LH. Got another LH question here. Secreted by the anterior pituitary. What do we think about that one? That's true. Secretion is inhibited following ovulation. Yeah, if you go back and look, the corpus luteum is going to be around after ovulation, and it's making estrogen, progesterone, and inhibin. All of those things have negative feedback on LH secretion. Directly stimulates granulosa cells to secrete estrogens. That's actually false. If you go back and look at the story, LH tells the thecal cells to make androgens. The granulosa cells then convert those into estrogens. Is it secreted in a burst in the middle of the ovarian cycle? Yep. And that's going to trigger ovulation. It triggers the corpus luteum to decay into the corpus albicans. That's that little white scar tissue structure. And hopefully you're thinking, no, that's not a function we learned about LH. And its secretion is stimulated by HCG to help maintain a pregnancy. No. HCG, human chorionic gonadotropin, stimulates the corpus luteum to keep making estrogen and progesterone, and also inhibin. And those three hormones actually have negative feedback against your luteinizing hormone. Okay, and again, I know some of this is pretty complex, and don't think the whole test is going to be quite this difficult, but I'm mainly focusing on these hormone stories and what's going on, especially over on the female side, because that's the most difficult part of the reproductive unit. Use your study guide and practice questions and homework questions to help you review. All right, which hormone directly stimulates thickening of the endometrium during the follicular phase of the ovarian cycle? So the key thing there is follicular phase. Which hormone during that first half of the ovarian cycle is stimulating this thickening to start taking place along the inside of the uterus? And if you go back and look, that's going to be estrogen, because the cells in the follicle, your granulosa cells, are secreting this estrogen, and that one of its targets is the inside lining of the uterus, and it stimulates it to become thicker, and blood vessels to grow into it, and those glands that are going to produce the nourishing fluids for an early embryo if it attaches. All of those things are happening due to estrogen. Progesterone gets made after ovulation during the luteal phase, when you have the corpus luteum around. That produces estrogen and progesterone, and the progesterone also plays important roles in making sure that those glands produce the nice nourishing fluid, and also prevents the uterus from contracting. That's another important function of progesterone. All right, so select each of the following that secretes estrogens. Again, there's more than one type of estrogen. All of them are chemically similar, but that's why you might see the term estrogens instead of estrogen at times. Ovarian follicles, yep. Corpus luteum, oh yes. Anterior pituitary, no. Granulosa cells, yeah, those are the cells in the follicle that produce the estrogen. They take androgens that were made by the thecal cells and convert them into estrogens. How about the inner cell mass of an early embryo? Nope. Placenta, yes, eventually the placenta that forms will take over production of estrogen and progesterone, and at that point you no longer need the corpus luteum. Be sure you go back and review that story. And how about the oogonia? Those are those stem cells in the ovaries that become primary oocytes, and no, they don't do any estrogen production. So our answers there were A, B, D, and F. All right, and so here's a diagram which is going to tell us a little bit more about this HCG hormone that we're talking about. And if you notice on the graph, we're looking at hormone levels of HCG, human chorionic gonadotropin, the estrogens, and the progesterone. And this is gestation in weeks, so we're talking about a pregnancy ongoing here. And notice for the first several weeks of a pregnancy, estrogen levels and progesterone levels are fairly low, but they're there at high enough levels to make sure that uterine lining doesn't slough off and cause a miscarriage to take place. And so early on over here, the placenta has not developed enough to be making estrogens and progesterone. Okay, so what structure is making the estrogens and the progesterone at that point? Hopefully you're thinking corpus luteum. Where is the corpus luteum? In the ovaries. Okay, so the early embryo produces this human chorionic gonadotropin, HCG. That travels to the target, which is the corpus luteum. That hormone is telling the corpus luteum, hey, you need to stay alive so that we don't slough off this early embryo that has attached to the inner lining of the uterus. We don't want to have a miscarriage here. So HCG is a signal from the early embryo to the corpus luteum, stay alive, keep making estrogen and progesterone. But then notice as you continue with the pregnancy, the estrogen levels and the progesterone levels surge, and that's because the placenta forms, and the placenta takes over producing these hormones. And by about 5 to 6 weeks into a pregnancy, you don't really need the corpus luteum making these anymore. It can go away. And so your human chorionic gonadotropin, the embryo gradually stops producing that hormone because it's not really needed anymore. And then the corpus luteum no longer has to stay alive and hang around because your placenta has taken over. All right, so what makes HCG? The early embryo. What is the target of HCG? The corpus luteum. Where is the target located? Where is the corpus luteum located? In the ovary that ovulated the secondary oocyte. What does that target? What does the corpus luteum produce? Estrogens and progesterone. Why is HCG not needed at high levels after 6 weeks? You kind of have a peak around the 6-week point. After that, you don't need it anymore. Corpus luteum doesn't need to stay alive. Why? Because the placenta starts making estrogens and progesterone for itself, so you no longer need those supplied from the corpus luteum. All right, multiple answer question. What would happen if an implanted embryo stopped secreting HCG within the first 6 weeks of pregnancy? Select all that apply. Do we think the pregnancy would terminate? Absolutely. No HCG, corpus luteum dies. If you're not making the estrogens and the progesterone, you're going to have a miscarriage is going to take place. The corpus luteum would live indefinitely. No, we just said it would die. FSH and LH levels would begin to rise. Yeah, most likely if the corpus luteum dies, it's producing the hormones that have negative feedback on FSH and LH release. So those levels would begin to rise. Menses, that's another term for menstruation. Would that occur? Yep. HCG drops down, estrogen and progesterone levels drop, menstruation or menses will take place. Progesterone levels would surge and trigger uterine contractions. No, if the corpus luteum dies, progesterone levels are going to drop. You are going to have uterine contractions. Progesterone inhibits uterine contractions. So if your progesterone levels surge, they would not trigger those contractions to take place. Estrogen levels would surge due to a lack of inhibin. No, the corpus luteum is making the estrogens. If it dies, you're not going to have a surge of them. A corpus albicans would appear in an ovary. Yes, the corpus albicans is the white structure. The corpus luteum, when it dies, it becomes a corpus albicans. All right, so that's A, C, D, and G. All right, go back and review what we discussed about birth control. Which of the following are actions, effects of hormonal forms of birth control? Select all that apply. So these are generally estrogen-progesterone combinations. Do they contain HCG to maintain the corpus luteum? No, they don't. HCG is produced by an embryo. Remember, that's your pregnancy hormone. That's the one that's detected in a home pregnancy test. Do they thicken the cervical mucus? Yes, that's one of the jobs of progesterone, is to thicken the cervical mucus. That creates a mucus plug around the cervix, and that's one of the things that's there to block sperm cells from entering the cervix and then moving on upward. from there. And also it's there to help prevent microorganisms from potentially moving into the uterus and possibly causing infections. Thickens the uterine lining. That one might be kind of tricky. You would think so, but if it actually, most hormonal forms of birth control, you have to have the right combination of estrogen and progesterone and the right amounts and produced in a sequence, like during a monthly cycle, to have a thick uterine lining. The uterine lining in females who are on hormonal forms of birth control is actually thinner. Do they inhibit secretion of FSH and LH? I hope so, because that's really the whole point. You don't want LH over here triggering ovulation. Do they trigger follicle maturation but not ovulation? No, they don't. Your FSH and your LH are suppressed due to negative feedback by those hormones. And so if your FSH is down, you're not going to have maturation of a follicle. Do they contain testosterone? I hope not. I don't think they would be very popular with females if the birth control combinations had testosterone in them and the females started sprouting beards and going bald and stuff like that. How about G? Prevents menses until the patient stops taking the hormones or takes fake pills for a series of days. Yes, that's true. Menses, again, is another term for menstruation. Vasectomies reduce the levels of testosterone in males and often require patients to take testosterone supplements. Hopefully you're thinking false. Again, I don't think a vasectomy would be very popular in the male population if it reduced your level of testosterone. Or testosterone is made by those interstitial cells in the testes, and testosterone is a hormone. Hormones enter the bloodstream. So the testes have little tiny blood vessels in them, just like any other part of the body. So when testosterone is made, some of it gets taken up into the blood immediately, and then it circulates throughout the body. So the testosterone does not need to travel through the vas deferens to get anywhere. It's going to enter the bloodstream. So a vasectomy will not affect testosterone levels. All right, and go back and review what we learned about the early stages of pregnancy. So we've got a uterus over here, fallopian tube. Here's an ovary. Here's the fimbriae of the fallopian tube. Here's all the things going on in the ovary. There's ovulation taking place, secondary oocyte. Starts traveling down the fallopian tube. Fertilization generally occurs within a fallopian tube, so there's little sperm cells. Remember, the single cell of a new individual is called a zygote, and that big gigantic cell will divide into, doesn't really change in volume very much at first. One cell becomes two becomes four. That's called the four-cell stage. Eventually, these cells keep dividing and form what's called a morula, which is a solid ball of cells. So that's what you looked like about 3 days after fertilization took place. That keeps traveling down the fallopian tube. This develops into this structure, which is basically like a hollow ball. And the cells that kind of form, if you think about this kind of being like a tennis ball with a mass on the inside of the tennis ball, that inner cell mass over here, those cells are going to become the embryo. And the cells of the tennis ball, those are called trophoblast cells. They become things like the amniotic membrane and the chorion that eventually contributes to the placenta. So they become the membranes that help support the embryo's survival. So there's your inner cell mass. And this is what implants along the endometrium, the inner lining of the uterus. So it actually embeds itself in there. And again, this is what's making HCG. So that HCG is traveling back up here and telling this corpus luteum over here to stay alive and keep making estrogen and progesterone. So that's what happens early. Now, we talked a little bit about this. We didn't get into embryology or the detailed development of the embryo all that much. But early in pregnancy, so here we're looking at like 14 to 15 days, 16 days, the embryo forms a disc-like structure. And here you're looking down on kind of on top of it, head and tail. And if you cut that open and looked at the side, kind of in a cross section over here, 14 to 15 days you have 2 layers. One layer is shown here in yellow and one in blue. Then about 16 days it divides into 3 layers, and those are called the germ layers. The inner one, the yellow one here, is called the endoderm. Middle is mesoderm. Outer layer is ectoderm. So that's what you look like at about 16 days. Okay, go back and look and review what we discussed. Okay, what are these cells in the ectoderm? What do they turn into? They're going to turn into certain structures. The endoderm, what do those cells turn into? What do these cells in the mesoderm eventually divide and specialize into? Easiest way to learn it is to learn, what does the ectoderm make? What does the endoderm make? And then everything else really comes from the mesoderm. So don't try to memorize all the things of the mesoderm. Just memorize what the ectoderm and the endoderm are responsible for, and then assume the rest of it comes from the mesoderm. And why is that important? It's because certain things that we get exposed to very early during embryonic development might impact the ectoderm or might impact the endoderm. And so you need to know, okay, if this chemical destroys cells in the ectoderm, what kind of birth defects might that embryo and eventually the fetus wind up having? Go back and review how labor takes place. So when you get to the end of the pregnancy, the uterus is very stretched. That stretching ultimately leads to oxytocin. So there are sensory nerve tissues in here that send signals up to the brain and trigger oxytocin to be secreted from the posterior pituitary. Estrogen has stimulated oxytocin receptors to develop on the uterus during the pregnancy. So oxytocin attaches to those receptors, stimulates the uterus to contract, stimulates the placenta to make prostaglandins, those signaling molecules we've talked about before. And these things cause a positive feedback to occur. The prostaglandins act on the wall of the uterus itself and make it contract more forcefully. And also these things stimulate more oxytocin to be released from the posterior pituitary, which just makes the contractions even more and more forceful. And so that's labor. Labor is a positive feedback process. It gets stronger and stronger and stronger until ultimately the fetus here is expelled out of the uterus and through the vagina and out into the world. All right, and I'll stop there. I didn't cover everything on the review. Again, I tried to home in or focus in on the key things. Use your review activities, of course, as always, to help you practice on Blackboard, and go back to your homework questions as well. And as always, make sure you know why the correct answers are the right ones on those review questions. It doesn't do you a lot of good to just try to memorize and regurgitate those questions, because your test questions are not going to be identical. If you do have any problems, you need help, again, I like you guys to use the discussion board so that everybody can see your questions, because it helps other people as well. Or you can contact me through a course message if you need private help. All right, good luck.